Hi, Professor Grunlin here. I have some introductory comments about the cardiovascular system and the heart most specifically. The cardiovascular system consists of the blood vessels, and the heart. In the broadest terms, the function of the system is to deliver nutrients, including oxygen, to the cells of the body and to take waste products, including carbon dioxide, away from the cells. In other videos, the more specific functions of the blood and the vessels will be described. This video, we're focusing on the heart. The heart is a relatively small, hollow organ. It has muscular walls. Its continuous beating, all day, all night, is essential for pumping that blood around your body. The heart is located in the thoracic cavity, nestled between the lungs. It's shaped like a blunt cone. The base of the cone is superior and the apex of the cone is inferior, pointing left. In fact, about 2/3 of the heart is actually on the left side of the midline of the body. On the external surfaces of the heart, you can see auricles and many great vessels entering and leaving the heart, and the coronary vessels clinging to the surface of the heart. Inside, the heart is divided into 4 chambers. Superior are the right and left atria, and inferior are the right and left ventricles. Flap-like valves separate the atria from the ventricles. These valves are called the atrioventricular valves, or the tricuspid on the right and the bicuspid or mitral on the left. These valves are attached by little cords to the papillary muscles inside the ventricles. When open, they allow blood to flow from the atria into the ventricles, but when closed, they prevent that backflow. Little cup-like pulmonary and aortic semilunar valves separate the ventricles from the great arteries. Imagine yourself as a red blood cell. You've just deposited your load of oxygen to a group of cells and picked up some carbon dioxide. You need to get rid of that CO2 and pick up more oxygen. You travel through the vessel system until you get to the great veins that return deoxygenated blood to the heart. If your journey was from the superior part of the body, you'll be in the superior vena cava. If you were in the inferior part, you'll be in the inferior vena cava. Or perhaps you were delivering oxygen to the heart itself in the coronary vessels. Then you're going to be returning via the coronary sinus. All that deoxygenated blood mixes in the right atrium, flows through the tricuspid valve into the right ventricle, then through the pulmonary semilunar valve to the pulmonary trunk, into the pulmonary arteries, and you're on your way to the lungs. In the alveoli of the lungs, oxygen will be loaded onto the hemoglobin. Carbon dioxide will be unloaded. That freshly oxygenated blood returns to the left atrium through the pulmonary veins. After passing through the bicuspid valve into the left ventricle, it will move through the aortic semilunar into the aorta. If you look close, you will notice that the walls of the left ventricles are the thickest part of the heart. There is a good reason for that. Contraction of the other chamber walls only needs to move blood a short distance, but contraction of that left ventricle has to be forceful enough to pump blood through the whole body. And speaking of contractions, in A&P 1, you learned about skeletal muscle physiology. You learned how the resting membrane potentials were generated and maintained, how opening sodium channels depolarized membranes, how that can sweep across a membrane via voltage-gated ion channels, and how that's followed by potassium channel opening and repolarization. You learned that depolarization resulted in the release of calcium into the cell, which allowed those power strokes, resulting in a muscle contraction. Recall that both skeletal muscle cells and cardiac muscle cells are both striated, and that striations are the result of a sarcomere structure. I hope at this point in the course you realize that if both skeletal muscle and cardiac muscle cells have similar structure, they're going to have similar physiology. And indeed, that is the case. There are some slight differences. For example, calcium influx required to start the power stroke cycle is usually from the smooth endoplasmic reticulum in the skeletal muscle, whereas in the cardiac, much of that calcium will actually be coming from outside the cell. And whereas we often use tetany in skeletal muscle cells to hold a contraction, for an obvious reason, you don't want that to happen in the heart. There's a longer refractory period preventing tetany in the heart. If it's been a while since you've studied skeletal muscle physiology, you may want to brush up on that. A large difference in the physiology is the source of the stimulation. Remember that skeletal muscle cells each needed to be innervated individually to stimulate the contraction. The heart, on contrast, has a built-in conduction system made of autorhythmic cardiac muscle cells. These cells spontaneously depolarize at regular intervals. These specialized cells form a network called the intrinsic conduction system. It works like this. The cells in the SA node depolarize, and gap junctions sweep that wave of depolarization through the atria towards the ventricles. Depolarization is followed by contraction of the atria walls. The contraction starts superior and sweeps inferior, pushing blood from the atria into the ventricles. Because there are no gap junctions between the cells in the atria and the cells in the ventricles, the depolarization does not pass into the ventricles. Rather, it has to move through the AV node to the atrioventricular bundle, then into the bundle branches, around the apex of the heart, and back up via the Purkinje fibers. That causes a delay so that the atria can contract fully before the ventricles start their contraction. The depolarization in the ventricles sweeps through the ventricles walls from the apex towards the atria. This depolarization is followed by the contraction of the ventricles, starting at the apex and pushing blood superior into the great vessels. That is, out the pulmonary trunk to the lungs and out the aorta to the body. When the ventricles are contracting, the pressure increase causes the tricuspid and bicuspid valves to close so that blood cannot go back up into those atria. The semilunar valves are open while the ventricles are contracting, but once they're finished, that pressure decreases in those ventricles, causing the semilunars to close so that blood can't backflow back into those ventricles. At that point, the tricuspid and bicuspid open so that blood can passively resume flow from the atria to the ventricles. This cycle repeats about 70 times per minute every day of your life. The heart sounds you hear through a stethoscope are actually the turbulence caused by the closing of those valves. Depolarization and repolarization is what's captured on an ECG. The P wave is the depolarization of the cells of the atria. The QRS is the depolarization of the ventricles and also the repolarization of the atria. And the T wave is the repolarization of the ventricles. Much information can be gained about the health of the cardiovascular system from the heart sounds and the EKG patterns. The number of times the heart beats per minute is called your heart rate, and the amount of blood forced out of the heart with each beat is called stroke volume. We will be getting into the factors that influence heart rate and stroke volume. But for right now, I just want to make you aware that heart rate times stroke volume is equal to cardiac output. Cardiac output equals the amount of blood ejected from the heart each minute, and the significance of that is that cardiac output affects blood pressure. If you increase cardiac output, blood pressure increases. If you decrease cardiac output, it decreases blood pressure. So we're going to be exploring the nervous system and endocrine system control over heart rate in order to change cardiac output and influence blood pressure. Well, that should get you started. Contact me with your questions as you're working through your objectives.